Right now, we are working on developing a human norovirus vaccine, which is about to enter into phase 1 clinical study next year. So our local authority also asked us, how does KAICO manage the quality of the silkworm? So one answer is that we use SPF-grade pupae from specialized cell culture facilities, strictly control diet quality, rearing conditions, environmental monitoring, and breeder documentation. So we collaborate closely with the PMDA, the Japan's Pharmaceuticals and Medical Device Agency, to establish the acceptance criteria and ensure alignment with pharmaceutical expectations in terms of the difference of each silkworm pupae.
For over 4,000 years, silkworms have spun silk that would eventually connect civilizations through ancient trade routes. But what if these creatures could do more than weave fabric? What if they could manufacture life-saving biologics? Today's guest, Masafumi Osawa from KAICO, is pioneering exactly that transformation. His team has developed the silkworm baculovirus expression platform that turns these living organisms into natural bioreactors. Join us as we explore how this technology is evolving from university research to GMP manufacturing reality and what it means for the future of protein production. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Masa, to the Smart Biotech Scientist. It's good to have you on today.
Thank you very much for having me today, David. I'm truly excited to be here.
Masa, share something that you believe about bioprocess development that most people disagree with.
Many people assume a living organism can't be a reliable or consistent biomanufacturing unit, especially when it comes to pharmaceutical-grade materials. But based on our work, I believe the opposite is true. A silkworm can be a highly powerful and surprisingly consistent bioreactor capable of producing a wide range of recombinant proteins, including vaccine antigens. There has never been a human medicine produced directly from a live silkworm. That's precisely why people question the quality and safety. Yet our experience is showing that silkworm-derived proteins can meet modern pharmaceutical expectations, and with the right controls, this platform can create new possibilities for biomanufacturing.
I'm looking forward to our conversation, Masa, to unpack this, to understand how you produce pharmaceuticals in silkworm. Before we do that, let's talk about yourself. Draw us into your story. What sparked your interest in biotech, and what were some pivotal moments that led you to your current role?
Unlike most of the listeners of Smart Biotech Scientists, my academic background is actually in cultural anthropology, not molecular biology or biochemistry. So during my university studies, I conducted fieldwork on Indonesian society as part of my research. Indonesia is a country rich in cultural diversity, but through my study, I also witnessed large disparities in access to healthcare, limited access to clean water, financial barriers to basic medicines, and gaps in essential health services. These experiences made me want to contribute to global health in a more structural way. And this led me to join Towa Pharmaceutical, a Japanese company specializing in orally disintegrating tablets. I began my career as a medical representative and later moved into international business development. I conducted market research in Taiwan and Mongolia, identified product market fits, and supported regulatory strategies for each region. It was fulfilling to watch new products reach patients and see their real-world impact. But during the COVID-19 pandemic, coincidentally around the time my child was born, I began re-evaluating my career path. I appreciated the importance of generics, but I also realized that generics only exist because someone first innovates and pushes the boundaries of drug development. I felt drawn toward innovation, toward work that might genuinely shift the trajectory of public health. Around that time, Japan was strengthening its focus on domestic vaccine development, and that was when I discovered KAICO and the Silkworm baculovirus protein expression platform. My first reaction was a mixture of shock, fascination, and respect. The idea that a silkworm, a small and fragile creature domesticated for thousands of years purely for silk, could produce complex recombinant proteins, normally requiring expensive bioreactors, was astonishing. I remember thinking, if this organism can produce such complex molecules, it could change the way we address disease. However, when I entered KAICO, I faced an immediate challenge. I had only worked with small molecule drugs and needed to learn the fundamentals of proteins, expression systems, and the differences between manufacturing platforms. Thankfully, with 90% of KAICO's employees coming from technical backgrounds, I was surrounded by researchers who generously supported my learning. This environment helped me rapidly bridge the gap. And interestingly, my anthropology background became a strength rather than a mismatch. Understanding how different societies collaborate, how decisions are made in different cultural contexts, and how technologies are adapted across regions became extremely valuable. And KAICO now works actively with international partners in Vietnam, Thailand, and Europe. And my ability to navigate cross-cultural communication has become central to my role. And today, as business development lead, I introduce KAICO's platform globally, support partners working with complex protein targets, promote our very first immune-enhancing product for pig, and build co-development alliances not only for vaccines but also for broader protein-based R&D programs. And looking back on joining KAICO felt like a natural extension of my original interest, connecting people, bridging cultures, and contributing to public health, this time through biotechnology.
I love listening and discovering your story, and I love seeing that it's not linear. You started at one end and now you're ended up in biotech. How fascinating is that? And also what resonated with me when you're saying, yeah, the non-biotech background, but actually this very experience you had, your studies, is actually a huge advantage. I love that. So tell us a bit more about how KAICO started as it was spinning off the university, and what is the vision behind your Silkworm platform? Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Okay, so KAICO was founded at Kyushu University, one of the Japan's leading institutions for entomology with over 100 years of history and more than 450 unique silkworm strains. Our CEO, Mr. Yamato, encountered the silkworm baculovirus expression system while studying in the MBA program. He was searching for dormant academic technologies with the commercial potential, and when he discovered this platform, he recognized its significance immediately. Together with Professor Kusakabe, the principal scientist behind the system, they founded KAICO in 2018. The company initially focused on two goals: developing recombinant research reagents derived from silkworms and collaborating with pharmaceutical companies to develop recombinant vaccine antigens and APIs. But beyond those objectives was a broader vision. If silkworm pupae could reliably express complex proteins at high yield, they could transform the landscape of biologics manufacturing. That vision became KAICO's mission, changing the world with silkworms.
So let's look at the silkworm more specifically. How do we inject a recombinant baculovirus into a silkworm? And how do you, quote unquote, culture your silkworm? Do you just let them grow and eat the mulberry leaves, or are they swimming in a bioreactor, or how does that work?
Let me walk you through the basics. So the silkworm system works through a straightforward but powerful mechanism. First, we designed the DNA sequence for the protein of interest. This sequence is inserted into a baculovirus vector that infects only silkworms. Once the recombinant virus is ready, we inject a small amount into the silkworm pupa. Over the next 4 to 5 days, the virus spreads throughout the pupa, infecting its cells. Each infected cell begins producing the target protein based on the inserted gene. During the metamorphosis stage, the pupa becomes a highly active biological environment with diverse cell types, abundant molecular chaperones, and physiological conditions that support the correct folding assembly of complex proteins. So practically speaking, the entire pupa functions as a compact, self-contained bioreactor with extremely high cellular density.
And these worms, where do you keep them? Are they in a container, or where do they live?
So we purchase all the silkworm cocoon from the local farmers or certain manufacturer, and each cocoon has a pupa. We first cut the cocoon and remove the cocoon and put the pupae in the container and put those in the refrigerator. In the refrigerator, they go hibernation, and we can keep them up to 1 or sometimes up to 2 months before injecting our baculovirus to them.
And how long does then the quote-unquote production process last? This is a few days, or is this weeks until you harvest?
So after inoculating our baculovirus, it takes just 4 or 5 days until the target protein fully expressed inside the body of protein. And after that, we purify it to get the reagent. So let's say 1 to 2 months if we have a right construct of the target protein.
And what I heard is that since the virus infects the entire worm, all different cells express your protein of interest. Is that correct?
Yes, you're correct.
Okay. Let's compare this now to more traditional platforms such as E. coli or mammalians, for instance, or conventional insect cell cultures. What are, I'd say, the key advantages of your system, or perhaps also some drawbacks versus the other systems?
So when comparing silkworms to other expression systems, several differences stand out. Compared to E. coli and yeast, silkworms offer more natural folding and more mammalian-like post-translational modifications. These characteristics are essential, especially important for structural proteins and multi-subunit complexes. Compared to insect cell lines like SF9 or Hi5, silkworms often provide higher yields, better folding integrity, and dramatically simple scale-out production. Insect cell lines require large bioreactors, expensive media, extensive facility infrastructure. Silkworms require none of those, and compared to CHO cells, the gold standard for therapeutic production, silkworms avoid the need for costly media, large-scale tanks, and significant water consumption. So silkworm-based production follows a fundamentally different philosophy. Instead of scaling up by building larger tanks, we scale out simply by increasing the number of pupae.
And I guess because you're scaling out and not up, you can much more quickly adapt to different demands, right? Because you're much more flexible.
Yes.
Now, something that comes to my mind, well, also that resonates with your first statement about what you think is different than perhaps from other people in our field. There are a lot of advantages by using a living organism. As you said, definitely the cost is much lower. That's one of them. And the drawback or potential drawbacks that comes to my mind is how do you manage the variability? Because if you, for instance, have a CHO cell line, that's a clonal cell line. So it's always the same clone. But I guess in your system, you have some genetic variability between one worm to another. How do you manage this?
Thank you very much. That's a very important question. Actually, right now, we are working on developing a human norovirus vaccine, which is about to enter into phase 1 clinical study next year. So our local authority also asked us, how does KAICO manage the quality of the silkworm? So one answer is that we use SPF-grade pupae from specialized cell culture facilities, strictly control diet quality, rearing conditions, environmental monitoring, and breeder documentation. So we collaborate closely with the PMDA, the Japanese Pharmaceuticals and Medical Device Agency, to establish the acceptance criteria and ensure alignment with pharmaceutical expectations in terms of the difference of each silkworm pupae.
And by doing this, you can manage the variability, so you can make sure that from one batch to another, you get the same product at the end of the day. Because in biologics, we say the process is the product. So imagine that in your system, this is true as well.
Yes, you're right.
Now, I've read on your website that you describe your silkworm pupa is equivalent to about 100 to about 1,000 mils of insect cell culture. Can you tell us a bit more how you came up with these numbers and what does that mean for the process economics? Does that mean that you can produce a lot more volume or more product on a smaller footprint?
So this number is not just an estimate, but it comes from a published comparative study titled Comparison of Recombinant Protein Expression in the Baculovirus System in Insect Cells and Silkworm. So in that study, 45 different recombinant proteins were expressed in SF9 cells, silkworm larvae, and silkworm pupae. When expression levels were normalized, the researchers found that a single pupa yields, on average, the equivalent recombinant protein amount produced by approximately 120 milliliter of SF9 culture, with some proteins reaching much higher equivalencies. This is where the 100 to 100 milliliter per pupa framework originates. So from a production economics perspective, this has important implications. As you know, conventional recombinant protein production requires large bioreactors, sterile media, and massive amount of water, followed by an extensive cleaning steps. And these processes contribute sustainability to both the environment, footprint, and operating cost. In contrast, silkworm pupae function as self-contained biological culture vessels. So they require no bioreactor, no large volumes of water, and no cleaning validation. The physiological environment is pre-assembled by nature, eliminating significant upstream costs. For developers like us, this also means that scaling is far easier instead of scaling up by building larger tanks, which adds engineering risk. You simply scale out by increasing the number of people, just like I mentioned earlier. This reduces infrastructure burden and supports longer-term cost efficiency. In the future, this advantage will make it easier to offer stable pricing and a consistent global supply.
Now, I'm curious, Masa, how do you do the downstream processes? Because once you finish your production run, these worms have expressed a certain amount of protein. And you mentioned that then you do the harvesting. So how does that work? And then how does the purification work? Is the purification very close to a traditional purification process we see with E. coli, for instance, or with yeast? Or are there some major differences?
The downstream purification process is similar to the conventional protein expression systems.
And how do you get the protein out of your worms? Is that similar to what you would do to E. coli, for instance? How do you, quote unquote, harvest your worms?
So after the target protein is fully expressed inside the body of pupa, we ultrasonicate the whole pupa body with a buffer. We ultracentrifugate the extracted form of the protein, and we run the chromatography. So basically the system is partially similar to the traditional way.
Yeah, I see. And that's also where I imagine you start applying clean room and close process conditions to make sure that at the end of the day, your product is sterile and safe to use, correct?
Yes, you're correct.
I'd like to touch upon the quality side of things because you mentioned glycosylation, which is an important part, especially for more complex molecules. Where do you see the limits of your platform versus to Cho? Are there certain molecules that are too complex to produce in worms, or you think we can produce pretty much any kind of molecule?
We can produce pretty much any kind of molecules. So far, one of our strongest technical advantage is that our consistent expression success. So across more than 130 protein expression projects, many of them challenging targets, we have observed expression in every case. So this includes membrane proteins, intrinsically disordered proteins, allergens, or multi-subunit proteins, large virus-like particles, even certain GPCRs. So many partners approach us after a successful attempt in E. coli or mammalian system. The silkworm pupa's physiological environment provides folding conditions that artificial bioreactors struggle to replicate.
As you're interacting now with health authorities, I imagine you have some very interesting conversations with them as it's a novel host, a novel way to produce pharmaceuticals. What are the unique GMP and regulatory challenges you have encountered so far with your living organism?
So because silkworms are living organisms, GMP considerations focus on raw material controls. So this kind of overlaps that what I mentioned earlier, but we use SPF grade pupae. So there is one certain facility, they only wear SPF grade super clean pupae for pharmaceutical use. They strictly control diet, what they eat, and rearing conditions, and also environmental monitoring and breeder documentation. So this is something that how to monitor the safety and quality is something that our conversation with our local authority is getting unique in that way.
That's it for part 1. We have explored how KAICO emerged from academic research and how silkworm pupae function as remarkably efficient bioreactors. Next time, we'll dive into the production economics, post-translational modifications, and KAICO's vaccine pipeline. If you're finding value in these conversations, please leave a review on Apple Podcasts or your preferred platform. It helps other biotech scientists like you discover these practical insights. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.